Hello friends. Nowadays, internet is full with news of coronavirus. But do you exactly know what is this virus? From where it comes? How it is so dangerous to human life? What it is doing inside our body? If your mind is buzzing with all these questions, then stay tuned till the end. Coronavirus is positive sense single-stranded RNA virus with the largest known viral RNA of 30 kb. It is formed from polyproteins which are dissociated by viral proteins to form various parts of the virus, like RNA-dependent RNA polymerase, ATPase helicase, surface proteins like hemagglutinin esterase protein, which helps the virus to attach and invade the host cell, S protein, which is the largest surface glycoprotein and also helps in the attachment of the virus to ACE2 receptors, an envelope protein, that is E protein, membrane protein, that is M protein, and nucleocapsid protein, that is N proteins. On and whole, a virus is a combination of lipids and proteins. Coronavirus are not new to humans. They are estimated to have existed millions of years ago in warm-blooded animals, example bats, as natural reservoir. They were detected in humans in late 60s and were known for causing common respiratory tract infections, and further studies suggested that these also originated from animal coronaviruses whose main reservoirs are bats, civets, and pigs. During those studies, these viruses were divided into two parts, that is group 1 and group 2, on the difference between the presence and absence of surface proteins. Here, this SARS-CoV-2 virus is genetically similar to SARS virus of 2002 and Middle East Respiratory Syndrome virus of 2012. This virus results in the disease COVID-19, which is nowadays a pandemic. It is said that this virus was first originated in a place Wuhan, Hubei in China in late 2019 and spread to over 180 countries by 1st of April. China reported to WHO on 31st December when number of deaths in China occurred because of severe pneumonia condition. Because of its widespread throughout the world, on January 7th, WHO named it as novel coronavirus, and this virus was renamed as SARS-CoV-2 by International Committee on Taxonomy of Viruses. China being the epicenter of this disease, there were highest number of cases in China which increased with time. During Chinese New Year, because of long traveling and very much traveling throughout the world, this virus easily spread throughout the continents, resulting in number of cases hike. Till 29th April, there are almost 3.12 million confirmed cases throughout the world of corona infected. Around 933,000 are recovered and around 2.17 thousand deaths have occurred because of this virus. India reported its first case on 30th January 2020, which was also a return of China in South India. Ministry of Health and Family Welfare confirmed around 31,332 cases of coronavirus till 29th of April, with recoveries around 7696 and number of death poll around 1007. They are considered to have originated from bats because of their genetic similarity around 96.2%. Most of the human coronaviruses have their origin in the bats only. MERS-CoV virus also emerged from bats but with the intermediate host of camel. It is also considered that sometimes rodents also act as reservoir. There are 7 types of coronaviruses which are seen in humans, out of which 4 produce mild symptoms and 3 produce severe symptoms. The severe symptom viruses are MERS-CoV of 2012, SARS-CoV of 2002 and 3, and SARS-CoV-2 of 2019. When we talk about rate of transmission, it depends on 2 factors. One is R0 and other is S1. R0 is the rate of reproduction, that is, ability of virus to replicate in the host cell or the person who is infected. Si refers to the series interval. It means time it takes to pass virus to another person who is healthy. R0 for this virus is considered to be 2 to 4 and Si is considered to be 4 days. If we take R0 as approximately 3, then it says that the infected person can transmit this virus to the next 3 persons after 4 days. Then these 3 will infect individually the other 3 persons, and these next 3 persons will infect individually more 3 persons, making it a chain of 12 days including infected persons around 40. Here is the graph which shows the relationship between R0 and infection. When there is exponential increase in the number of patients with time, R0 is said to be more than 1. When there is a stable line between number of patients and time, then it is said to be R0 is equal to 1. And when there is exponential increase with a steep fall, then it is termed as R0 less than 1. It is seen that this virus transmits through respiratory droplet infections, through coughing, sneezing, or even while having conversations. It also spreads with fomites. These are the materials which are likely to carry infectious material like clothes, curtains, cardboards, plastics, gloves, glass, with a different time of keeping the infectious agent. This infection can also spread to aerosols, that is the suspension of tiny particles or droplets in the air. And this can carry infection for around 3 hours in the air. Virus can travel more than 25 feet in less than a second. This virus needs ACE2 receptors for its attachment to the human body. Thus, it can infect various organs including lungs, heart, intestine, kidney, and liver. Lungs being the most affected organ because of plenty of ACE2 receptors in the alveoli cells. Here we have discussed pathophysiology of this virus inside the host cell. What is the process that goes inside the cell? As you can see, here is a coronavirus, SARS-CoV-2 with its proteins. With the help of S protein and hemagglutinin proteases protein, it tries to attach to ACE2 receptors present in the wall of the type 2 pneumocytes which are present in the lungs. With the help of these proteins, this virus manages to go inside the host cell, where with the process of endocytosis, this cell dissolves, leaving its RNA behind into the cytoplasm. This RNA binds to the ribosomes present freely in the cytoplasm or on the rough endoplasmic reticulum. With the help of these ribosomes, this RNA manages to form polypeptides, that is proteins, polyproteins. And with the help of RNA-dependent RNA polymerase, this viral RNA manages to form more of the copies of new viral RNA. These polyproteins or polypeptides are dissociated into proteins by the help of proteases. As there is presence of proteins and the viral RNA, these both can be used as raw material to make new viruses. Both these things accumulate in the Golgi apparatus, and there they are assembled into a capsid, forming a new virus. Number of new viruses formed then are released into the cell tissues with the process of exocytosis. Because of use of this cell to form new viruses, this cell ultimately dies. During all this process, what is the fate that is going secondary? Here, infected cell or the dead cell releases interferons to initiate inflammatory response or immune response. Interferons signal the healthy cells of the alveoli to release viropeptides to kill the virus. It also increases the release of cytokines to signal macrophages to start their inflammatory action. Macrophages start releasing interleukin-1, interleukin-6, interleukin-8, tonoferon, and interferon gamma, out of which interleukin-1 and interleukin-6 sometimes move out of cell to the endothelium of the capillaries, passing through the alveoli, resulting in the constriction of the cell and creating gaps within the wall. Because of these gaps, there is leakage of fluid into the interstitial tissue. This fluid sometimes enters the alveolar cells and results in alveolar edema. Because of presence of fluid in the interstitial space and also in the alveolar cells, there occurs a thick layer between the alveolar cells and the capillary endothelium, resulting in poor gas exchange. Because of poor gas exchange, there is fall in the pressure of oxygen. Less oxygen inside the capillaries results in hypoxemia. Because of less oxygen inside the capillaries, lungs start struggling to respire more. More struggling results in more damage to the cells, resulting in more poor diffusion of the air. Ultimately, there is increase in saturation of carbon dioxide, resulting in hypercarbia. In hypercarbia condition, carbolic acid converts and releases protons. Because of presence of protons, there is fall in the pH of the blood, resulting in acidosis. Over time, these also act on the endothelium of the vessel, from where WBCs manages to move into the alveolar cells. Their neutrophils and WBCs and macrophages release reactive oxygen species and proteases. Macrophages released interleukin-8. All these factors try to signal other cells and result in the damage of type 1 and type 2 both the cells. Type 1 cells help in the gas exchange and type 2 helps in the formation of surfactant. Because of reduced gas exchange and increased alveolar collapse, there occurs more hypoxemia and resulting in breathlessness to the patient. It is said it also increases the activity of anticoagulants and increases the activity of procoagulants. Because of increase in the activity of procoagulants and decrease in the activity of anticoagulants, it can result in DIC. It is also said that sometimes, because of more secretion of factors like leukotrienes, prostaglandins, bradykinin, there occurs stimulation of vagus nerve which results in cough and bronchoconstriction. This is all what happens inside the cell. This happening inside the alveolar cells is also called cytokine storm. The cytokine storm can manage to damage number of cells of the number of organs, and that too essential organs of the body including heart. It can also result in the formation of embolism, resulting in more worse features. This is all. We will discuss about its signs and symptoms in our next video.